do a land acknowledgement. I want to recognize that we live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the truth of the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Toronto, which is covered by Toronto Purchase Treaty Number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. But I know that many of our members live in Vancouver, which is part of the unceded ancestral and unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And this is brought to you by the Women's Health Research Cluster. You might even notice that we have a new logo. We have over 570 members now, so that's a little bit of an error there. So it's free to join, so please spread the word about our community. We actually represent 29 countries across the world. At the moment, we run a number of events, including this Women's Health Seminar Series. I'm busy populating that for next year, so that announcement should come up pretty soon. We have a number of conferences that we also have done. A lot of them are online in our workshops, as well as a trainee research presentation series. We also have some travel awards. There'll be a new announcement coming up really soon, so please do participate that. If you can, you have to be a member to apply for a trainee travel award, and it can be from anywhere globally in the world that you're applying, and we have a really good success rate, so please do take advantage of that. And we have a podcast, a blog, as well as a seminar series. And obviously, thank you to all of our sponsors. So without further ado, and one sponsor that's missing that should be on there is Manulife. So we really appreciate Manulife's support. Now I'm going to stop sharing my screen. And while I'm stopping sharing, Dr. Lai will share his screen and I will read out his bio and fun fact. And don't forget to unmute yourself, Dr. Lai, or else it'll be a very short presentation. Dr. Lai he has both an MD and a PhD, which, it's hard enough getting one of those degrees. So I'm always think kudos to people that get both of those degrees. That's pretty amazing. He's a psychiatrist and senior scientist at the Center for Addiction and Mental Health in Toronto. He's also an associate professor and co-chair of the Advisory Council for Equity, Diversity, Inclusion, Indigeneity, and Accessibility and Clinical Chair, CLARE, in the Department of Psychiatry, and a graduate faculty at the Institute of Medical Science, and also at the Department of Psychology at the University of Toronto. He's an Honorary Visiting Fellow at the Department of Psychiatry, University of Cambridge, and an Adjunct Associate Professor and Attending Psychiatrist at the National Taiwan University Hospital. He's an INSAR Fellow and an editor of the journals Autism and Molecular Autism. He's also a Sex and Gender Science Chair and that's a chair that was awarded by the Canadian Institutes of Health Research. And Dr. Lai's work, which you'll hear about, is on how sex and gender-related factors act as modulating mechanisms for the presentation and adaptation, clinical recognition, neurobiology, and the etiologies of neurodevelopmental and co-occurring mental health conditions. His fun fact, when Dr. Lai was a psychiatry resident, he was also the concert band director for the high school from which he graduated in Taiwan. And allegedly, there's video of this on the internet. So if you're really interested, you might be able to suss it out. I haven't watched it yet, but I can't wait to. So without further ado, Dr. Lai.
Thank you so much, Lisa. Hear me okay? Great. So thank you, Lisa, for the kind introduction, and thank you everyone for giving me the chance to be here and sharing some thoughts and perspectives about this research related to how we integrate sex and gender mechanisms into autism neuroscience. What I'm hoping to do today is to walk us through what we know at this moment about how we integrate sex and gender perspectives to the autism spectrum. As many of you know, autism is a neurodevelopmental condition historically considered as a disorder, but now recognized as part of neurodiversity. It has a prevalence rate about 1%, based on medical diagnosis around the world. It does occur in people who are assigned male or assigned female at birth, and these are people who you might be able to recognize on the top left, who is Stone Triplet, who is actually the very first person who has been recognized in the English literature as being autistic, described by Leo Connor, who recently passed away in the age of, I think, close to 90. And then we have this famous Temple Grandin, a professor in animal science, who had initially had language delay, who are diagnosed with autism later on, but then who went on to develop a great amount of literature, like scientific contribution to animal science, but also to the advocacy of autism. We also get people like our colleague Fleischmann in our own country, who is a augmented and alternative communication user, and who has also been a TV show host, and also people like Stefan Witscher, who is a savant who can actually who has this like photographic memory, and this is when Stefan Witscher like drew the landscape of London as part of say his artistic work. We do get people around the world across different race, ethnicity, and also sex and gender who's on the autism spectrum. But historically, our understanding of autism is actually mostly still based on what we understand about autism in male individuals. So if you look at the textbook description of autism, they usually cite this number of a male to female ratio, about 4 or 5 to 1. This is one of the most comprehensive meta-analysis on the rate of autism diagnosis across epidemiological studies. They generally have a rate summarized as 4 to 4.2 to 1, male to female. If you stratify those studies by how the study is ascertained, you get some differences. So if you only look at those studies who collect information from existing clinical or educational database, you get some rate of higher, like 4.56 to 1. But if you look at active ascertainment, which means that you go into the population without assuming people are autistic, and you did a population-based survey, epidemiological research, you get a lower rate. So essentially, the idea is that there could be some female individuals who are assigned at birth who were not recognized by existing educational or clinical databases. And things have been improving over the years, especially if you look at the US rates, national surveillance rate, the rate is actually moving towards this, between 3 to 4 to 1 ratio. But there are still this ratio that's still biased towards male assigned at birth who are more likely to be autistic or being diagnosed with autism. And this is a complicated scenario because historically, but also in current literature, we know that not only autism, but across neurodevelopmental conditions, there seems to be having this increased likelihood for male individuals to be diagnosed with autism. But the literature is actually quite limited by how they define sex and gender, because they usually do not really have the capacity to delineate the different factors related to the mechanism side of things. For example, they only have a male-female label to give us this ratio, but we don't know exactly how these different sex attributes or the gender attributes play a role in this ratio. But these mechanisms are important because they could actually give us a range of implications about how things can be interpreted. And I'm going to touch upon a few today, mostly from the neuroscientific perspective. Some of them are about what we know about the brain in terms of how they differ by sex and possibly by gender. We know quite little about that at the moment, but also how we can make inferences based on what we know about the brain to the emergence of these neurodevelopmental conditions, which is what we called whether they are moderators or whether they could actually be potential mediators in terms of some of the sex and gender related attributes coming to the emergence of autism. So I have to refer to this slide every time from Lisa's group, which is basically telling us and showing us how we have improved over the past decade, how much still needs to be done in terms of making sure we have a good representation and balance of the study. So this is basically a study showing us how in the neuroscientific literature, what are the studies that actually use both sexes as their sample in understanding cases, in understanding the scientific topics of interest. Things have improved over time, but the optimal design to understand sex differences or sex-related mechanisms, and I would argue also for gender, is slowly improving, but still not too much. And really, there's really the minority of them have the optimal design to talk about sex-related factors or differences. And you can see many of the studies are enriched in males, and also many of them are actually, like very little of them are actually focusing on female individuals. This profile is not too different in the autism field. So this is a like descriptive summary of what things were like about 5-6 years ago. So I looked at a few different meta-analysis or large-scale studies, and basically what they showed was that, you know, back at that time, most of these studies had a quite balanced male to female ratio. So, for example, TOS fMRI studies 10 years ago have a 15:1 male-female ratio. In about 2016, in terms of also say brain organization studies, you have a 9:1 ratio, and then things have improved over time, but still, like even in large-scale behavioral studies, about 5 years, then still like about 5:1 male to female ratio. So there is actually quite the underrepresentation of female individuals in autism research, especially in autism neuroscientific research. My lab member, Kelly Mu, had conducted a wonderful, very comprehensive systematic review on the literature about any studies that actually has the capability of looking into sex and gender-related factors in autism, especially about brain structure and function. So Kelly surveyed the whole literature between 2000 and 2021 about autism and sex and gender, and looked at the subset that addressed issues related to brain function and structure, primarily neuroimaging studies. But I want to draw your attention to this exclusion, because among these number of studies, we excluded about 758 for the reason that they did not look into sex and gender, or they merely control sex and gender as a variable, which basically means you do not have the ability to understand what's happening related to potential sex and gender effects. So eventually, we only get about 69 studies that properly at least look at sex by diagnosis injection or looking at sex stratification analysis. So that's still a minority of all the studies. But even within that 69, you get this interesting mix in terms of description. So most of the study use sex label, and they provide when they provide the definition of that, they will provide it correctly, but rarely do they highlight how that designation was defined. And many studies use gender, but they're actually not giving any definition, and they may not really fit into what we understand about gender nowadays. So there's a huge gap in terms of how we understand sex and gender in this particular literature. And these are the studies already looking into sex and gender effects. So I just want to point out, like anything I'm going to share later on are confined and constrained by these limitations in the literature. So before I dive into the most updated one, I thought it would be helpful to share with you like my own journey of learning, because this particular field has evolved quite a bit. I started my grad study in 2008. And at the time when I started my grad study, I initially wanted to do something about the social neuroscience and connectivity theory related to autism. But when I get to Cambridge, I got this chance to be supervised by the amazing Simon Baron-Cohen, who has contributed a lot to understanding of sex and autism. And Simon asked me this question: actually, there are very little study on what happens with autistic females. So are you interested in looking at that aspect, and do already have interest in sex and gender separately to autism? And that makes a perfect match for my study. So I'm also lucky enough that at the time, there is an ongoing study led by Professor Declan Murphy in King's College London, which is called the MRC Autism Imaging Multi-Center Study, looking at understanding neurobiological features of autism in autistic adults. And that study was primarily recruiting male individuals at the time. So this is a meta-analysis actually showing the landscape about less than 15 years ago, a meta-analysis about the neuroanatomical studies of autism. As you can see, most of the study sample sizes are fairly small based on current standard. and they are primarily with males assigned at birth individuals. So you really know very, very little about what happens with autistic females at the time. So I took on that opportunity to say, okay, let's look at what happens in autistic females. And at the time, we're still following that rule of thumb of having a group of about 30 individuals. Then you get to understand more about what's happening there. Of course, now we know that's not appropriate sample size, not powered enough in current literature, but I just want to show you what we got at the time. So in order to solve this issue, I thought, let's look at autistic individuals, non-autistic individuals, male assigned at birth and female assigned birth, using a 2x2 factorial design. And at the time, there were two theories that's more directly addressing what we may understand about autism and what we call sex differential likelihood. And I apologize that some of the terms used in these slides are not really updated to current standard based on our community-based knowledge nowadays. I'm still using them because I'm citing the original studies. So on the right, this is what we call multi-threshold liability model. Basically, it's a model to explain the factors contributing to the emergence of autism should be distributed in the general population. But there could be factors that pushing the male population more likely to more likely to have people who cross the threshold of being clinically autistic. And there could be some mechanisms pulling the female assigned at birth population away from that threshold, so you get less people who are diagnosed autistic. And these factors could be genetic, it could be other factors, and I'll talk about that later on. But that's what we call multi-threshold liability model. At the time, or actually before that, Simon had this idea at the cognitive level, trying to explain what autistic cognition might look like by presumed, on average, sex differences in cognitive profiles. And what Simon proposed was basically follow what Hans Asperger proposed almost 80 years ago, to say that they proposed that the autistic cognitive profile is an extreme form of the male-female differences on average in the population at the cognitive level. This theory has actually been led into a lot of misconception and really exaggeration of the implications that resulted in a lot of inequity in terms of autistic females not being properly recognized clinically. So there could be assumptions based on the name of the theory to say that, okay, autistic females must look like autistic males, or even more so for them to be autistic. So if you don't look like autistic males, you're not autistic, right? And that's actually not what this theory initially proposed to do, but it was misinterpreted or interpreted by people later on. But I want to point out that even the theory is a little bit outdated. It did have a implication about aligning the access of male-female differences on average and the mechanisms underlying that to the access of autism, non-autism differences. Basically, it's the emergence of autism. So aligning these two axes or the mechanism aligned with them on the same direction actually is in line with the multi-threshold liability model, because this is a mechanistic model for you to understand why there are more male assigned births being diagnosed autistic. It could be related to some sex differentiation mechanisms that's happened early in life as part of the explanation. So I just want to point out the possible misconception of the theory back in the days, but also how it's actually in line with the genetic understanding or proposal about emergence of autism. And I tried to figure out how I can address that possible predictions at the brain level. So essentially, this is the best diagram I could come up with at the time. It's still very complicated. But essentially, if you think about a group-based approach, this is male non-autistic people, this is female non-autistic people, and this is male autistic people, and this is female autistic people. So there are two different research questions, actually. One is that, is sex moderating the neurobiology of autism, right? And to answer that question, essentially, you just compare the effect of autism in the brain or other places in the male and in female. That's essentially sex stratified analysis, and how these two look alike or not alike. If they are not alike, there are significant interactions in a 2x2 factorial design. If they are alike, there are no interactions and there are significant main effects. So that's the basic idea. If you want to align, the dimension of sex differentiation to the dimension of autism emergence, you actually examine how these two effects overlap, right? So the typical sex differences, excess, and the autism in male individuals, that would address the predictions from the EMB theory in male, and you can do the same in female. So that's how I figured to be an interesting question to look at. And I'm showing this raw output at the time, SPM glass brain. Some of you may know that. And I was looking at pairwise comparisons after a factorial design interaction. And this struck me at the time when I visualized them from a quite linear threshold. So you can see this is white matter volumetric differences. And this is the effect of autism in female individuals. And this is the effect of sex differences in the non-autistic individuals. And there are some similarities in terms of the differences. You can actually eyeball them already. And I was quite struck. I was checking the membership and making sure I'm not doing things incorrectly. But that did stimulate my thinking about, okay, so how can we address that through this particular diagram? So this became a paper that was published more than 10 years ago, and there are two questions to be answered. One is really, are the neuroanatomy characteristics different in autistic males versus females by appropriately considering typical sex differences? And you would basically examine diagnosis by sex interaction, and we did find diagnosis by sex interactions in certain white matter areas. But secondly, I tried to quantify the extent of overlap between the typical male-female differences and the autism effect between autistic and non-autistic people in males and in females separately. So the prediction of the extreme male brain theory at the brain level would be that the effect of autism would overlap with the effect of male-female differences. And this is on the y-axis, this is the percent of overlap. The dotted line of this black regions are basically permutation-based overlap generated by random. And then we look through a range of different thresholds of this, basically the T-map of the group-wise comparisons. And what we showed was that across gray and white matter, in that small sample of 30 per group, we did show significant overlap between the effect of autism in female individuals and the male-female differences in the non-autistic population, basically fitting the criteria or the prediction of the EMB theory, but that wasn't the case for male individuals. So essentially, you have to look at that separately for male-female individuals, and then it seems to be the case that this kind of effect overlap in neurophenotype seems to be more evident in autistic females, but not so much in autistic males in that sample at the time. The mechanisms underlying that, we don't know. This is really a cross-sectional study looking at the emergence, looking at the overlap, and maybe we can make some inferences based on that. So based on that, I also thought about how we interpret these different angles of sex and gender issues in autism. And basically what I've talked about is this understanding sex and gender independent and dependent characteristics in the brain. And they may be able to inform about the general models of etiology, which is basically the multi-threshold liability model. And then the extreme male brain theory model would be one of the potential mechanisms that's specifically linking autism to sex and gender differentiation. So things have evolved quite a lot. So I want to quickly walk us through what has been, what we have known so far over 10 years, more than 10 years from that time. So there are a range of things to understand sex and gender-based moderation. So this is basically sex and gender dependency of autism effect. So this is a great collaboration with Rana DiMartino and led by Dori Flores and Jose Filho in Child Mind Institute. And Dori and Jose looked at the functional MRI dataset using 3 datasets, the ABIDE, which is the large-scale open access dataset, amalgamated by Adriana, and then also look at the EU Ames LEAP dataset, and also the GENDAR dataset by Kevin Pelfrey. So what we did was that looking at a large data set, that's available, like well matched in young people, and looking at across resting state functional connectivity metrics, what metrics showed a sex dependency of autism effect. So essentially looking at the significant regions in the brain that has a diagnosis by sex interaction. So we had this dorsolateral occipital cortex of voxel-mirrored homotopic connectivity, basically showing that in the autistic female population, there is a reduced metric of that, meaning actually lower bilateral functional coordination in this region. And then typically, this is what people do in brain-wise analysis, that you look at what the cluster means, and this is a cluster that is related to visual processing, oculomotor processing, and cognitive flexibility. And this cluster in female individuals also showed a correlation that the higher autistic traits measured by ADOS was corresponding to lower metrics on the VMHC, which is on the same direction of the diagnostic group differences. So this is one example of how people trying to understand localized diagnostic effect that's dependent on one sex or gender. In the structural aspect, there's another example. This is led by Sashi Bedford, which was at the time at Mala Chakravarti's lab in McGill. So Sashi looked at, again, across data set that we put together, and Sashi did a sex stratified analysis looking at cortical thickness differences. And what was found was that, the regions or the clusters showed significant diagnostic effects were actually quite different between the male strata and the female strata. And if you look at the effect size maps, they also look quite different. If you look at the correlation maps between ADOS score, which is autistic trait scores, and the correlation to the brain metric in terms of the neuroanatomical metrics, they also show quite different patterns across the two sex strata, though we do not get significant diagnosis by sex interaction in this analysis. So although you do not get a significant localized region that's passing the threshold for interaction, if you look at the overall brain pattern, you just see kind of like distribution differences, right? So that's actually highlighting the importance of looking at a sex stratified analysis. Based on that idea, we're thinking about, okay, so how do we address that properly? So there are two models explaining, really trying to extrapolate the multi-threshold liability model, that looking at how is sex modulating the autism effect. And if you look at the historical model, which is basically saying that you need to have more brain changes for you to become autistic, then the reasonable assumption would be that the regions involved would be the same one, right? So you can think about, let's assume if you need like 1 unit change in amygdala volume for you to be autistic in male individuals, and following the quantitative sex modulation model, you actually need 3 unit changes in amygdala volume for you to be autistic, but they need to be at the same brain region for them to happen, right? It's the same brain region involvement. So that's a simple quantitative sex modulation model. It could be the other scenario, actually, which is like the brain changes corresponding to autism are actually quite different. It's not like overlapping between the two. So they are qualitative different. They could involve different regions, or they could be different directions of changes. So what we did was that we took the PON dataset led by Evdokia Augusto and Jason Lurch alongside Margot Taylor's dataset as SIGKIT, all within one site, 2 scanner versions. And we're trying to assess these 2 potential different models by looking at how much of the brain regions fit into this prediction, which we call a sign concordance model, and which of the regions fit into this prediction, which is essentially a spatial correlation between the autism effect in males and females. Which is what we call spatial correlation model. And that's corresponding to these different predictions. And what we found surprisingly is that there's no regions in the brain that fit into this particular quantitative sex modulation model. We didn't see a brain region that's consistently showing related to autism and larger effect sizes in females. We actually see quite different distribution of autism effect, especially in cortical curvature across threshold for between the two sexes. So again, showing that the brain correlates are divergent, likely by sex when it comes to autism, when it comes to the correlation with autism. And these are two studies that I'm not involved in, but actually point to the same directions of effects. So this is a study led by Elodie Couvet in Sven Bolt's group in Sweden. And in a population-based twin sample study, the RAS study enriched with people who are diagnosed with autism, what they looked at is for a comparable increase of autistic traits, like 1 unit change of autistic traits, what are the corresponding brain anatomical changes in the female and the male individual separately? And they use this typical coloring, but like the red regions are those regions corresponding to 1 unit of autistic trait changes in females, and the blue regions are the one for males. And you can see there are more regions involved in the brain for the females, but also there are different regions than those in the male ones. So again, that's showing there are likely more changes, but they actually happen in different brain regions. And another study, probably the most important longitudinal study, the Autism Phenome Project, led by David Amaral and Christine Wu Nordahl in UC Davis Mind Institute, tracked people from age of 2 to 4 all the way to their adolescence. And this is a study led by Josh Lee, who looked at amygdala-connected network in the anatomical data set using multivariate distance metric regression. And you can see that on the left-hand side, these are the regions that's corresponding to the sex effects, the diagnosis effect, differently for the male sex, which is denoted in the green bars, and the female sex, which is denoted in the pink bar. And you can see that those regions contributing, or the effect size of the regions contributing to these diagnostic effects separately for the two sexes are actually quite different. And their dissimilarity coefficient, which is showing how similar they are, is quite low. So again, suggesting that the diagnostic effect in the amygdala-related networks are quite different in the male versus female individuals. And that also shows a trend of the pattern in the trajectory, so across time points as well. So again, independent data set showing this possible sex differential characteristics of autism in the brain. So the latest study efforts that, again, led by Sashi Bedford and in collaboration with Richard Bettelheim in Cambridge, we put together basically the largest possible data set across ABIDE, POND, Healthy Brain Network, GENDAR, ADHD 200, UK MRC AIMS, and UCSD Biomarkers of Autism Project led by Eric Kirshen, basically trying to recapitulate what we have done before, and using specifically this brain charting idea, and some of you might have read this idea or this study, very important one by Richard Bettelheim, published in Nature 2 years ago, is basically the structural brain growth chart by sex and by age, and using that to be more sensitive to detecting autism-related differences, this time across autism and ADHD cohorts. So what we see is that if you look at sex by diagnosis and interaction effect, you do see, again, significant interactions this time in volume for the temporal regions, but you don't really see that effect in ADHD. But what's more important is that we also conducted a sex stratified analysis and looking at, again, like how similar or different the maps of the diagnostic effect maps look like in the male strata versus the female strata. And this time we conducted a spin test, which is a similarity test. Essentially, the more significant, the more they look alike this time. So what we found was that if you look at cortical thickness, that you do see a quite significant similarity in terms of autism effect. But that wasn't there for cortical volume and wasn't there for service area either, especially for service area, they're actually quite not alike in terms of autism effect across the brain in the two strata. But when it comes to ADHD effect, they're actually quite alike across the male and female individuals. So we see some specific autism-related sex-moderated brain features. So finally, what we do know about gender? We don't, to be honest. There isn't really good data about gender characterization in those data sets so far. There's just one sex/gender label that we have so far. The attempt to get into this field is really spearheaded by John Strang at Children's National in Washington, D.C. And John had a unique data set with a binary youth. So these are all transgender diverse youth who are all binary. Basically, they are either trans men or trans women. And John had them also, some of them having autistic diagnosis, some of them are subclinical for autism. And they looked at this functional connectivity based on default network. And what was found, drawing your attention to this one, is that you've got this significant effect of trans young men larger than trans young women in terms of default mode networks connectivity, which was known for the sex assigned at birth effect already. Like we do know in large scale studies that there is a higher on average within default mode connectivity in birth assigned female than males. Again, in this particular design, we cannot delineate sex at birth effect versus gender effect because these are all trans youth, and there isn't a cisgender group that could be provided for comparison. So I just want to point this out to say that, we're working on this, but we're not there. We don't really know about what's happening in terms of gender identity or gender role or gender stereotype effect in the brain and how that's intertwined with sex at birth effects. So finally, I want to draw your attention to a little bit of this convergence idea and this the initial predictions about the extreme male brain theory and how that plays a role currently in terms of aligning the autism, non-autism access to the male-female differences access. So one latest development, again, led by Dory Flores and Han Peng, and I'm very proud to be part of this team, is to, again, pull together large-scale data sets and trying to ask the question, if sex differentiation mechanisms as presented in the brain with brain morphology as the outcome is overlapping with the mechanisms related to autism, then how would that intersect with the effect of autism in the brain presented by neuroanatomical features at the outcome? So the prediction would be that if you are managing to create a model that can predict the sex label of the brain in the non-autistic population, and if the mechanisms of sex differentiation and the mechanism of autism overlap to some extent, then the prediction would be that that model, that sex prediction model, would be more likely to misclassify autistic females than autistic males. So they're more likely to misclassify autistic females as males than autistic males. But in the female comparison, that model would be more likely to misclassify autistic females than non-autistic females. And then in the males, the model would actually be more likely to correctly classify autistic males, right? Because that male-female differences as presented in the brain would overlap with the autism effect in the brain in the same direction. So if they do overlap, then the prediction would be that the prediction model will work better in autistic individuals. So what was done was that Heng and Dory took the UK Bell Bank data set through a deep neural network model. It's called a simple fully convolutional network model. Deep learning model. And they created a model that can actually predict the sex label of the brain imaging, T1 weighted image with very high accuracy. They conducted transfer learning to use that model to predict the abide data set. And what they found was that basically all three hypotheses hold. So on this graph, you will see that if this is basically the differences of the prediction accuracy at the group level of the first group versus the second group. So hypothesis one predict that the prediction accuracy will be lower for autistic females than males. So it's actually lower than 0. And this will be no differences for neurotypical male and females. So basically they predicted poorly, the model predicted more poorly in autistic females than autistic males. They predicted more poorly for autistic females compared to non-autistic females, but they predicted better for autistic males compared to non-autistic males. And then they replicated the findings in the LEAP, EUAIMS LEAP dataset, finding the same. But that wasn't the case in ADHD samples. If you go through everything the same way, it's not shown in the ADHD sample. And this prediction accuracy differences is most prominent in the younger windows than the older ones. So it's actually more prominent in the earlier ages than the later ages. So, like indirectly addressing this might be something more related to something early mechanisms than the later mechanisms. But again, this is not suggesting autistic people all have extreme male brain in a literal way, right? Because if you look at the individual prediction distribution, you can see that most of the female individuals are correctly predicted as female individuals by the model, autistic or non-autistic. It's just that you have this heavy tail of like inaccurate or misprediction in the autistic females groups, more likely than the non-autistic groups. But the majority of females are predicted as females, and the majority of males are predicted as males. So the extreme male brain prediction shouldn't be interpreted in the literal way that they have to show a extreme. I mean, because sex differences in the brain is already with small effect sizes anyway. It's more about what the possible convergence or overlapping mechanisms might look like and how that could turn out to be at the level of brain anatomical differences if you follow that prediction. So this is like the latest development that's pointing to the possibility of a convergence of the mechanism of the two. And in this paper, there are also some other speculations by doing this with the brain gene expression maps, and I really highly encourage you to look at that. But that leads us to the question of what are the possible molecular mechanisms leading to that? And my friend Donald Whirling has done a great job trying to summarize this for us. So if you look at the possible mechanisms, it's really about what are the overlaps between sex differential mechanisms and the autism-related mechanisms. So there are two possibilities, right? So one is that sex differential gene regulation has a direct effect in the genetic background of autism, but it could also be that there's a downstream effect that's actually overlapping with each other. So Donald looked at the brain gene expression data set that was available at the time and Donald found that basically there's no evidence supporting a direct effect. So sex differential mechanisms did not directly affect autism related genes per se, but there is a enrichment in autism upregulated gene modules, especially in the immune microglia, so immune modules, that also happen to be enriched in male individuals than female individuals in the general population. So there's a sex differentiation or sex differential gene expression already in typical development, and basically males are enriched with these immune mechanisms, especially early in life, prenatally, and that's overlapping with the upregulated genetic modules in the autistic brain. And again, these data are really quite confined by the small numbers of this, because these are basically brain data that come out of post-mortem tissues. So we really don't know exactly what happened in autistic females, for example. These are still majority autistic males, but at least it provided some indirect evidence of the alignment or convergence of these two mechanisms. And they did summarize this fairly well and similar to what I have described, but in a better way, that if you want to understand sex differences, you look at them directly. But if you want to understand the convergence of sex differential mechanisms to a condition, here autism, you do have to compare, basically the sex differences in the typical population and then how that correlate with the other axis, which is the so-called case control differences. And again, they found that in a larger data set, the modules that's basically more enriched in typical development in male than females are also the modules that is having a upregulation in autistic brains, which is essentially the immune and glial related mechanisms. And that fits very well, actually, with the animal work. And I always refer back to this great work from Peck McCarthy, basically showing us that historically, in rodents, people talk about sexually dimorphic nuclei and about the size differences of that. But also importantly, there is a cellular differences that the microglial cells are in different forms, enriched in different forms in male mice versus female mice, and that cellular mechanisms is actually a immune-mediated, a PGE-mediated mechanism, but that's regulated by estradiol, which is a masculinizing hormone. So there is a link between the neurohormonal mechanisms to the immune mechanisms, and that's probably setting the stage of what we call a higher likelihood for male individuals to be exposed to immune, more active immune mechanisms. So if there's further factors facilitating or enriching that immune mechanisms, that could lead to higher likelihood of the agent or the individuals becoming neurodevelopmental or having autistic diagnosis later on. And this is my study, but this is a potential biological mechanism that could lead into the human studies that we're talking about. And that's really the molecular mechanism that's aligned the phenotypic observations in the brain that I have referred to in terms of the convergence of the male-female on average differences and the autistic, non-autistic differences in males and in females separately. But we do have a lot to learn, and these are only in 10-ish years time that we have progressed a lot with more data, but we still have a lot to learn. So I just want to summarize a few lessons that I learned. So first of all, we have to be aware and we have to know that yes, historically, there isn't a good measurement about sex versus gender separately, but we probably have to consider that carefully moving forward. They're not the same. They're both multi-component constructs, and they're not always binary, and many of them are actually not binary, and many of them are multi-categorical. Some are dimensional. Each factors related to sex and gender may have different distribution characteristics that we have to keep in mind. We still have a lack of data from non-male individuals, and we still have a lack of data about gender, not to say a lack of data about different genders in terms of autism-related characteristics, clinically and in the brain as well. And still, most of the research to date are still coming from this all this research I presented still based on this one single binary variable, which is at best a proxy measure. So moving forward, definitely increasing sample diversity and representation to ensure that we have sufficient assigned female at birth individuals in studies across the board, and then including different gender identity and expression groups that can actually enrich our understanding of gender effect on top of sex effects. And we need to make sure we measure these complexities to capture more variances, and we need to measure the components that are relevant, and then we can model them separately. So I just want to sum up with a few slides, and this is a brilliant graph made by my student Kelly, who basically outlined the vicious cycle of excluding some people, especially female individuals, from the beginning, and then further excluding them because of co-occurring conditions, and then you generated a knowledge lens that's already quite male biased, and you use that lens to feed into future study, then you create a vicious cycle of further exclusion. So if you're not attending to intersectionality, then we're actually not being able to capture the full picture. And then there's very little study looking at contextual effects in environments. So I don't have the chance to talk about this area of masking camouflaging in individuals on the spectrum, but I want to point out this particular finding that we had. So this is again from that data set I collected with colleagues in my grad school. This is a functional data set that there's a paradigm looking at thinking about self versus others, and then you can see that there is a distribution differences in neuroactivation in VMPFC, which is related to self-other processing. So autistic males had a hypoactivation compared to non-autistic males, but autistic females had a hyperactivation compared to non-autistic females. And we also had a proxy measure of camouflaging or masking, which turned out to be that the higher measure of that, the higher neuroactivation of this, right? So you can think about that as two ways, because it could be that you can argue that autistic females or females in general are equipped to be more able to think about self versus others, so more likely to compare themselves and then to mask and camouflage in social situations. You could also argue the other way around, that because of gender socialization or other pressure, that they are forced to camouflage more, so they are forced to think more about the differences between self and others, and making the neuroactivation higher during this particular task. But these are all the intricate like relations between the context and the brain. And we really cannot delineate them very clearly without longitudinal study, intervention studies, or other experimental designs. And these are only cross-sectional correlations. And we have to attend to the context. And then we have to think about sex versus gender. I think that's also important to delineate the different factors when we think about mechanisms. So with that, I want to bring your attention to a recent group paper really highlighting the areas of improvement that's needed for clinical, but also for research related to sex and gender, especially attention to assigned female at birth. So the group has highlighted a few different areas that require more attention in the future, especially including much more female individuals in autism neuroscience and clinical neuroscience studies together. So highly recommended for us to take a look at this one. So I want to thank you all and all the funders for the research and my group. And I'm very happy to take questions and discuss with all of you.
Well, thank you very much. For those of you online, if you, I don't want you to unmute and clap, but if you do the little emoji, that'd be great, which I can see a bunch of the little emoji claps already. So thank you so much for that. That was a force. And you kind of, my mind is a little exploding. I'm thinking about like because sometimes I will use different terms, but I love the like qualitative versus quantitative thoughts about how sex and autism might be related or not related. And I have questions, but I'm sure that other people do. So if you have one, please put your hand up and you can unmute, or you can put it in the chat, and I'm happy to read it out as well. I just don't want to always be about me asking the questions. So if somebody, I'll just talk a little slower, but I will kick us off if I don't see anyone putting up their hands yet. You can see me going very slowly. I tweeted, of course, some of it out. I didn't tweet out the paper that was in revision. That was really, really interesting as well. Like, I think what you found in terms of the in revision paper was cortical thickness was similar between males and females with autism, but not volume or surface area. And I think that's really interesting. So I thought about a couple of things. One is, and I didn't write that one down, the first question I wanted to ask you, if it's manifesting differently, I'm wondering about the timing as well. Like, does the timing, because all of these are probably obviously at one stationary time point. I think there was one study that you showed that was longitudinal. So is there some timing differences, and does that suggest that different treatment strategies might need to be used for males versus females with autism? Or is it, do you see what I'm saying?
Yeah, I mean, this is really like a tough and $1 million question.
Sorry.
And first of all, I mean, this is a great one. And I mean, Sashi really did a great job in aggregating all these differences, all these data sets, and like conduct all the analysis. So, kudos to Sashi. I think the findings, there are cross-sectional findings, right? So this is the limitation. And actually, in the past 10 years, we benefit a lot from these multi-center studies, but we're actually repeating things on these multi-center studies, right? So this is still, the best we know about this is still from that 400-ish autistic females in the 10 years who contributed a lot. But still, that's a small sample compared to the general population at large. So first of all, there's a limitation on that across a wide age range. It's cross-sectional study. But I do agree that when we're looking at morphology, that the surface area versus cortical thickness differences did imply that the mechanisms could be different. And that implication there would be that if there's any sex-moderated mechanisms leading to autism, that's what we call more qualitatively different, that could be through the different mechanisms related to the expansion of the cortex versus the cortical thickness metric, like the underlying mechanisms to that. But the problem then is that we really don't have the data to speak to that, because everything we know now is basically from children to adulthood. We don't really have any prenatal data. We don't really have any early childhood data to begin with. So at best, it's a speculation that there could be mechanisms that's linked to the two different metrics that's more sex dependent in this case. Now, when it comes to neurobiologically informed support for autistic individuals and how that could be tailored to sex, I think I would approach that more from a clinical needs perspective, because I think that's actually more practical, because that's more proximal.
And makes more sense, yeah.
It makes more, yeah, it's really harder to trace back all the way to the very early ages, especially we're not having the data on that early ages to start with. So I think more studies are now like doing more sex stratification analysis, especially for intervention ones, and they trying to tailor the intervention by the findings there. I think to me that's making more sense in terms of creating more sex informed support.
Yeah, I mean, I think there's so many things that, thinking about the cross-sectional versus longitudinal questions that could come up there. And Rosie, I'm going to come to you in just a second. Because I'm now wondering, because you brought up Peg McCarthy's work too, like, is there timing differences in terms of when a person could be more susceptible to autism during development? So if there is like well, I just saw something crazy, like if the mom took Tylenol or something, like, but and like just I know there's nothing to this, so I don't want to spread that. If we think of it as something that might have happened at a certain time, is there a different critical time for a male versus a female that sort of changes the trajectory of cortical formation, and that might be contributing. So like maybe the susceptible time in a male assigned to birth is at a different time than it is in female. So I think that's another, like, I mean, I don't know if there's any research on that.
Yeah, not that I'm aware of. It's a very difficult one on the other hand, right? So because, for example, the perturbance of neuroinflammation that contributes to like neurodevelopmental outcomes, it's not contributing to autism per se. It's also contributing to a lot of neurodevelopmental conditions,
exactly.
right? So from that perspective, some people would argue that if you can try to optimize neurodevelopmental outcomes by removing as much as language delay or intellectual development delay as possible, that would be important. But there will be people have a lot of concern about whether you should actually do something to erase autism to start with, right? So if you're doing something like that, so there's a concern and even debates on that end,
right?
But theoretically speaking,
theoretically
speaking, yeah, if the sex differentiation mechanisms do play a role in modulating the probability of one become autistic or neurodevelopmentally disabled at some point, then that would be probably the timing, right? But the tricky thing is that, for example, if you look at the sex differential gene expression, study, that's like later, like close to birth, right? So it's throughout,
right?
So I think there's lots of complexity and timing issues, and we don't really know. And my own approach is that let's start with the clinical and the current needs. I think that's more immediate.
Yeah. I agree, and I have a bazillion other questions, but I live in the same city that you do, and the same institute. So I will take it over to Rosie. Rosie, do you want to ask a question, or do you want me to read it out? Do you want to ask it? Just unmute yourself and.
Yeah, sure. Can you hear me okay?
Yes.
Yes, thanks for a really great presentation. My question is just around how we go about including autistic women in research, because of the barriers that they face, first of all, to diagnosis within the community, and then also when you do then rely on screening measures such as the ADOS, that leads to and contributes to underrepresentation within research because it excludes more autistic women than autistic men. Yeah, so are there kind of current best practices? I know maybe there isn't a great answer, but
it's a brilliant question. I think it's actually very necessary for all of us researchers to make sure that when we design studies or think about studies, that we work together with autistic individuals and autistic women included. And that's what we are trying to do in our program as well. So I think from the researcher's perspective, ensuring that we take participatory designs in mind and apply that as much as possible. I think that's the principle. Then in the context of community practice, I think there are increasing understanding of how things can how things can be biased from different angles, and instruments and their interpretation could be one of them. But there's beyond. So I think more conversation with community practitioners is also what we're trying to do, and to bring attention to the possible different presentations and the biases that could exist, to trying to reduce that barrier as much as possible. And again, like for clinicians in the group, we're not diagnosing people based on one instrument, right? So Kathy Lord and Summer Bishop actually highlighted that very clearly for all of us. I mean, ADOS is a tool that's useful. It's not the sole instrument to make a diagnosis. It's part of the information. So the most important thing is like how clinicians make a proper comprehensive diagnosis based on all the information that is available to them.
Oh, that's a really great point. Autism is not my field of expertise, so I think about, but I do think about other things. I do think a lot about what have we been missing because we have spent so much attention on the males. And I think in autism research, it probably is the really well highlighted that because of this sort of inherent bias, because of the prevalence rates, that we have missed some certain things. But it's also important to recognize there are many tools out there, and how can we develop the best tools out there. And diversity matters even in our tools, is what I was going to say. So with that, we have reached 1 p.m. For those trainees in the audience, there is a mentorship session afterwards, so I encourage you to stay on. But for the rest of us, if you would just join me in thanking Dr. Lai again for an amazing, mind-blowing talk. I have to listen to it again and think about a lot of things, and I want to buy you lunch so I can talk to you more. So thank you so much. And then